So this is core component mock, and this is mock number 2, and we're on to the essay. What we're going to do is just give you an overview of the sort of knowledge you need, not actually write the essay for you. So molecules and ions have different ways that they can cross the cell membrane into a cell. As soon as you see that, you should be possibly thinking of a table, and that compares different types of transport. So for example, types of transport, and then essentially if you can remember sort of 3 things about them, whether they, so is it a gradient? So gradient tells about that. Tell us about whether they are active, so need ATP, and tell us whether they need a protein. And for your kind of types of transport, so for example, diffusion, osmosis, facilitated diffusion, active transport, and endoexocytosis. Then you can get your sort of thoughts organised. Gradient, so diffusion, it goes from high to low concentration. Osmosis, high water potential to low water potential. Facilitated diffusion, high to low. Active transport, low to high. Endo and exocytosis, well, it doesn't really have a gradient there. Are these active processes? Diffusion, no. Osmosis, it's not active, so that's passive. So passive. Facilitated diffusion, type of diffusion, so again passive. Active transport is active, needs ATP. Endo and exocytosis, well, it requires energy to do that, with all the membranes and so on. Do we need a protein for diffusion across a membrane? Nope. Osmosis, a little bit of a tricky one. We sort of say no, but there are kind of little aquaporins, little proteins, so again, not in the big sort of proteins that we associate with some of the others. So maybe a little bit of confusion there. Facilitated diffusion, well, the protein facilitates the diffusion, so we need a protein. Active transport, we need a protein. And endo and exocytosis, we can't transport those through proteins. It's too big a process. We need budding and vesicles and things like that. So one way of tackling this essay would be to give essentially that table, but written out in full. And then we've got our examples, and we've got, so oxygen, we'd then say, okay, well, oxygen, that must be by diffusion, and we know that anyway. We can say oxygen, it's a small molecule that can fit through gaps in the phospholipid bilayer. Is it affected by cyanide? No. So not affected by cyanide, not affected by cyanide. So it's definitely diffusion, our passive diffusion. What does, well, we'll discuss what cyanide does. It says it's a respiratory inhibitor. So cyanide is providing that ATP. So, sorry, respiration, particularly aerobic respiration, provides that ATP, and much more about that in the second year. And if you have cyanide there, it's a respiratory inhibitor, so it will inhibit respiration, so we'll get no ATP, and so anything that's active on this list will be able to happen or happen less. So oxygens are a straightforward start, small enough to fit through the phospholipid bilayer, straight diffusion. We have got vitamin D. Well, vitamin D, rate of uptake with or without cyanide is exactly the same, so we're going to go for diffusion again. Not particularly because it's small, but you should know, some things can diffuse across a membrane. So if we've got our structure phospholipid bilayer, we've got our fatty acid tail, so this is hydrophobic, our fatty acid tails, and we've got hydrophilic phosphate heads. So fatty acid tails and phosphate, and this is our phospholipid bilayer. So vitamin D, that can get through, because it can get through this layer because it is lipid soluble, and so it can diffuse. And again, we're going to say it's definitely diffusion because not affected by cyanide, so not affected by cyanide. Then we get to glucose. You know a lot about glucose. You know that it's a polar molecule, which means it's got regions of small charge on it. And we have got, without cyanide, 1, with cyanide about 1/3 of that. So cyanide reduces this movement of glucose. So we're going to suggest it's active transport, requiring a protein that is powered by ATP, and cyanide will prevent much of that from happening. So our first suggestion for glucose is its active transport, as the rate affected by cyanide. And we'd tell the story about cyanide respiration producing ATP. But we notice that it goes down, but it doesn't go down to 0. So some glucose will still move even without ATP. So our second suggestion would be, well, actually it's facilitated diffusion. So this doesn't need ATP, but it does need a protein with a hydrophilic lining. So on this diagram here, we might have protein with which this hydrophilic lining here, and so glucose could go through. So some facilitated diffusion, still some uptake even with cyanide. Then we move on to fructose. So fructose, so data from the table, 0.29, 0.29. So we are not going to go for active transport, but fructose, we know, is very much like glucose. The structure is a polar molecule, so we'll need to travel by facilitated diffusion down a gradient. So I'm going to put facilitated diffusion. Cyanide has no effect. And the last one, a bit of a tricky one to answer here, are proteins. Well, our proteins are pretty big. Our proteins are generally too big to go through another protein. If I was to draw a kind of protein on this diagram, that sort of size, then you can, well, it's not going to fit through that little gap there. We need to move this membrane around a protein, fuse with the membrane, and move it through, in bulk. And that's the way that it happens. So proteins too large to go through the membrane, and so we would describe it being engulfed in vesicles and endocytosis. Is it endo or exo? It's into a cell, so it'd be endocytosis. So we'd say engulfed in vesicles, endocytosis. And is the rate affected? Well, the rate, we had a very very low rate of uptake. It actually goes down to 0 with cyanide. So affected by cyanide, so must be active. And have we covered everything that we need? It says discuss the methods of uptake for each molecule. So we discussed each molecule, the method of uptake, and the reasons for any differences in uptake. So we've discussed that. So we've done all of that. In the top band of marks, sort of 7, 8, 9, we're looking for the types of transport correctly linked to the right molecule, and kind of sound reasoning, a bit like what we've done there. In the middle, we might have explained these different types of transport, but not necessarily got all of them right, and attached the right molecule to the right type of transport. In the bottom band, sort of 1 to 3, we're giving some facts about some forms of transport. We might have said, diffusion of oxygen, but then got some of the others wrong. So, clear gaps in our knowledge there. Quite a lot of work actually for 9 marks there.